think this video will play for you, so let's play it together. Maybe? No. Well, that's a bummer. Let's see if I can fix that. This is what I would be doing with you anyway, so let's see if I can fix this video for you. Oh, it's because it's not recognized as a video for this one. All right, apologies to that. You can open this thing up in your Canvas, on your Canvas page. You can go to the videos section and then find the video on phagocytosis, and it will show you this process wherein phagocytic cells approach bacteria, viruses, or other foreign substances, and they engulf and destroy this. But instead of the video, let's just continue walking our way through. Of course, if you're looking at this from home, you can pause this video at any time and go and look up these other videos that are embedded in this, well, should be embedded in this PowerPoint, to look and see what that looks like. But let's just draw a picture and describe the activities of this. So phagocytic cells, what they do, we've already talked about the one that's in most abundance, always circulating in the tissue, which are neutrophils, right? 60% of our circulating white blood cells are these seek and destroy guys. They survey the tissue, and most importantly, that neutrophils, right? There are different locations for these ones. The neutrophils are in the blood. They circulate through the blood, and they're doing surveillance, but they're really just looking for any signs of inflammation or injury in any of the regions of the body, as detected by chemicals that enter the blood in a very particular location. And when they receive that chemical signal, they'll stop where they are and they'll migrate from the blood into the tissue. But we do have resident tissue macrophages, as we mentioned before. There are other tissue resident cells too that can help elicit or initiate this response for the inflammatory response. But if there are foreign microbes, injured or dead tissue, particulate matter, right, their job is to ingest them and eliminate them through very particular digestive enzymes. And some of these phagocytic cells, in particular macrophages, dendritic cells, and even B cells, and we'll talk about that in our next chapter, but these guys are also responsible for pulling out antigens, which are small fragments or parts of different proteins or components of that ingested material, and present those antigens on the surface, and downstream they'll call for help. And the help that we're talking about is the adaptive immune system, right? The T and B cell lymphocyte response. So let's see how this whole process works, the mechanisms behind phagocytosis. So phagocytic cells must first get to the injured or invading tissue, right? The injured tissue where you might have invading microbes or dead or damaged tissue. And this process is through chemotaxis. If you recall, chemotaxis is the concept of diffusion following a chemical gradient, or movement towards or away from a chemical gradient, right? So if we had a bacterial cell that's damaging some of our tissue, so let's say this is one of our epithelial tissues and that big bacteria got inside of it, holy cow, it's hard to draw with these things. That host cell is going to be sending out very particular chemicals that are designed to help elicit a host immune response. And these chemicals will diffuse away from that injured, right? So in high abundance here, from that source of injured tissue, and these chemicals will diffuse out away from this thing. And if we have our circulation right here, our capillary system, and we have our neutrophils or white blood cells rolling through this area, there we go. If they detect this chemical gradient, they will migrate through the capillary system into the tissue and go in the direction of that chemical gradient. Wow, it's like slow motion here. And once they arrive on scene, they will probably find those microorganisms, right? So if this is bad guy, this is bacteria. And that phagocytic cell, once it arrives on scene, remember it has those toll-like receptors that are on the surface, and if that toll-like receptor is engaged, it binds that bacterial cell, it will begin this process of phagocytosis. So recognition and attachment through that toll-like receptor binding to a host cell, binding to a foreign cell. This is followed by engulfment, fusion of the phagosome with the lysosome, destruction, and exocytosis. So let's look at a picture of this first before we go any further. This is from either your current or last edition of the textbook. Let's kind of walk our way through this step by step. Again, these phagocytic cells, in this case, we are looking at a multi-lobed monocyte or a macrophage that's arriving on scene. Remember, neutrophils are the first to come, but macrophages and dendritic cells come downstream. So this is the arrival of that, or the migration of this white blood cell to the source of injured tissue or invading bacteria through these chemotactic factors that are released usually by injured tissue. Sometimes bacteria themselves can be secreting particular metabolites that are also chemotactic factors. Chemotactic factors, this term here, right here, chemotactic factors, that means factors or chemicals that create a chemical gradient for chemotaxis, right? So this is what the actual chemical is that the white blood cell is migrating towards in chemotaxis. So these guys arrive on scene, and we'll kind of toggle back in. I don't know if I'll have to do that. I think that we could just look at this image here. So these white blood cells will arrive on scene. Once they get here, there must be recognition and attachment. So before we even get to this point of engulfment, please note that these pattern or pathogen-associated molecular patterns must have a physical interaction, and I'm going to add this guy in here, must have a physical interaction. Let's make it... See if I can do it on here instead. Nope. Okay, so these PAMPs must bind to surface pattern recognition receptors like those toll-like receptors we mentioned. And so there must be an actual engagement between PAMPs and I'm going to put my colon here, and TLRs, right? Toll-like receptor, physical interaction binding to those pathogen-associated molecular patterns or microbial-associated molecular patterns. Once this binding has happened, that will elicit a response inside the nucleus in the cell, and a physical change will happen. This binding will initiate phagocytosis, which is engulfment first. And remember that they're still physically bound to these pattern recognition receptors. Oops. All right. So step number 2 is the engulfment of that phagocytic cell. Let's see if I can change. So here's step number 2, the engulfment of these pathogens or bacteria into this phagocytic cell, forming something called a phagosome. The phagosome is that initial engulfed compartment. And inside this phagosome, you will have, really nothing happens right away, but these extra granules that we have all over the place, these extra endosomes are called lysosomes. These guys have tons of digestive enzymes inside, like lysozyme, as you know of, but other important molecules too. So step number 3 would be the fusion of the lysosomes with the phagosome to form something called a phagolysosome. This phagolysosome is where we have all of this digestion. So there's tons of antimicrobial chemicals that are secreted in here. Here's sequestration of iron. There's hypochloric acid, hydrogen peroxide, right? This is hydroxyl free radicals that are generated from the combination of all of these chemicals together. There's lysozyme, there's other digestive enzymes, and their job is just to emulsify that bacteria or virus that's inside of that phagocytic cell. Once you have complete digestion, then this whole thing will spill itself out to the external environment called exocytosis. And this is all phagocytic debris. This usually elicits a second call for phagocytic cells. So if this is the first time around, this would be like a neutrophil spitting out all those contents. But if this is a macrophage, dendritic cell, or if this is a neutrophil, they're spitting out the contents, creating pustulation, which also drives the attraction of these downstream macrophages and dendritic cells that can pick up all that debris and then take it to our lymphatic system, where it can call for help. This is from your current textbook. This is showing you the same process. I just don't like it quite as much. But again, there's the bacterial cell. First, we have to have chemotaxis, right? So phagocytes migrate to the area of inflammation by being attracted to chemical gradients that are released by the injured tissue. Once they arrive, they'll bind to any bacterial cell through those pattern recognition receptors, like toll-like receptors that are on the surface of these cells, and those recognize particular patterns on the surface of bacterial cells. This binding will elicit engulfment or endocytosis. This is into a first compartment called a phagosome. You can see here we have these extra endosomal vesicles called lysosomes that are filled with digestive enzymes. These lysosomes will fuse with the phagosome to form a phagolysosome, and here we have digestion and destruction of bacterial cells. Here's a nice list of many of the enzymes that are involved in here. So lysozyme we know of. DNase and RNases are enzymes that degrade both DNA and RNA. We've heard of proteases before. These are enzymes that break down proteins. Peroxidases, we've heard of this with hydrogen peroxide, or like catalases, a peroxidase, a type of peroxidase, but in this case it's eliciting or creating these hydroxyl free radicals. There are reactive oxygen species. We've mentioned this term before, including that superoxide anion that we've talked about, hydrogen peroxide we've talked about. There's singlet oxygen and hydroxyl anion that can be generated in this phagolysosome vesicle. And this is extremely toxic to bacterial cells. There's almost no bacteria that can evade this host defense once that lysosome has fused. And so what you're left with is just a bunch of residual debris that's mostly bacterial components, including a lot of these enzymes and reactive oxygen species that are then released by the cell. So this is exocytosis or elimination. And here's a whole discussion of some of these processes here. I'm not going to ask you to remember all of these very particular key reactive oxygen species, but it would be good for you to know that reactive oxygen species, or ROSs, are part of that phagolysosome and destruction of bacterial and viral cells. Just so you know that there are many bacteria who have evolved defenses against this process, where they actually allow themselves to be taken up. Salmonella is a great example. They love to be inside macrophages, so they allow themselves this whole process. They're recognized by our phagocytic cells. They're engulfed into this phagosome, and then they secrete their own enzymes that halt the process right here. So they prevent the fusion of the lysosomes to the phagosome, and essentially they get a free ride inside that macrophage in these nice protected vesicles inside that cell. So some bacteria have evolved specific host defense responses to protect against our own host defense system. What are the types of phagocytic cells? We wrote a bunch of these on the board on Thursday, but neutrophils, as we know, are the key phagocytic cells involved in the process of tissue repair or tissue defense, surveying any injured tissue. They arrive first on scene. There are rapid responders, very, very effective in their phagocytic action, but they have a really short lifespan. Monocytes, which are in circulation, can differentiate into macrophages and dendritic cells. So once they get inside the tissue, and I think I'm missing it. There we go. Monocytes, once they get inside the tissue, can differentiate into macrophages or dendritic cells. These guys, macrophages, are in the tissue themselves. So we drew a picture of how these guys and mast cells are surveying the tissue themselves and can respond right away if there are any foreign invaders at that site. And these are some of the receptors that they have, some of the receptors that they have and kind of their functions. So macrophages can go and activate T cells downstream. This is a super cool picture. This is actually using something called scanning electron microscopy, where you can really get a nice, very, very high resolution, high magnification image of what's happening. And this is actually a group of bacterial cells in the pink being engulfed by the fluffing membrane of a macrophage right here. This image is from your book. This is a pretty decent image to show you where these phagocytic cells might be in tissue. So if you have tissue resident cells, you'll find your macrophages there. Neutrophils are not tissue resident. They are primarily in circulation and only arrive in the tissue when there is an injury response. Dendritic cells can be in there. And then what you have here is just a network of connective tissue, like the reticular fibers that connect all of these different tissues together. And as you maybe would expect, you have a large concentration of these in these areas where our lymph nodes are, so we can have a connection between our tissue and our lymphatic system. We're not going to go over this discussion question here, but we can talk about when I get back into lecture. The question is, macrophages perform the final job of removing tissue debris and other products of infection. Indicate some of the possible effects when these scavengers cannot successfully complete the work of phagocytosis. And again, this does happen with very key types of pathogens that physically halt the function of these phagocytic cells. So I do want to mention also, while we're here, before I move on into talking about the other components of the inflammatory system, which we'll get back to there too. But let's switch over for just a second. We'll see if this drawing is any better. So I do want to mention that during this process of phagocytosis, and we'll draw it in blue. So here is our phagocytic cell. We'll draw a nice, I think we will draw. No, we're still not drawing yet. Bear with me. All right, I'll draw in blue. And let's use, okay, that looks like it's working. Try again. All right, so here's a picture of our phagocytic cell with our TLR. These guys are responsible for binding to bacteria. Let's put that in a different color, like this. So here's our bacteria with their pattern recognition receptors like lipopolysaccharide all on the surface of the cell. And when that bacteria binds to the toll-like receptor, where we have, again, we have PAMP binds to TLR. Right here. This binding then will trigger, so let me move back over here, the engulfment of this whole complex. I promise, guys, I do know what I want to do. Maybe, maybe not. The engulfment of this whole process into oh, that's not going to work for me. Oh, maybe I'll go back to here. Oh my gosh, I'm stuck. Okay, yeah, here we go. So we have this engulfment of everything here. Sorry about that. Where all of this stuff enters into the cell here. This moves into a phagosome, where we still have our bacterial guys in here. We have no digestion yet. This is the phagosome. This fuses with lysosomes here. So this is a lysosome that has lots of digestive enzymes in here, and this complex forms a phagolysosome, which is where we have digestion, right? So enzyme and ROS, reactive oxygen species, break up that bacteria or pathogen into all these little pieces in the phagolysosome. This phagolysosome is where all of that enzyme digestion occurs. So as I just showed you this picture, the last step here would be exocytosis. Exocytosis, but with some exception. Our neutrophils, actually all of our cells will do this exocytosis, but our antigen-presenting cells, so we wrote this on the board the last time, and I'll add this to our abbreviations, APCs, antigen-presenting cells, they actually do one more thing. They grab a hold of these antigens, and with some of them, they stick them on the surface of their cells. And we'll talk about specifically how this process occurs in our next chapter. But for now, just note that there are very specific surface receptors that our antigen-presenting cells have, and their job is to take those foreign proteins, like little bits and pieces of that bacterial cell, and they present them on the surface. All of these little phagocytic debris, anything that's there. And so they become these little flagships, right? And then they will take these guys, and once this process begins, these guys will migrate. Once this process begins, these antigen-presenting cells, APCs, will migrate to the lymph nodes. Lymph nodes are these meetup spots we have between these guys, these antigen-presenting cells, and our lymphocytes. So their job is to go to our lymph nodes, where they will present antigen, or little pieces of that bacterial cell, which we call AG, to our lymphocytes. Okay, and we'll talk in a lot of detail about how this happens in chapter 13. So the antigens are these little components of our invading bacterial or viral cells or particles that have been digested and broken down, and these little pieces are now called antigens. All right, these guys right here. Antigen. So let's add a couple of words to our abbreviation list as we go through this. We did talk about APCs before. These are antigen-presenting cells. And the two most classic ones are dendritic cells and macrophages. Dendritic cells are the primary ones responsible for activating our third line of defense. Macrophages activate usually our memory cells, but nonetheless, they're both very important. B cells are also in this category, but we'll bring that back up again later. So what is an AG? This is what I say is an antigen, which is small parts or fragments of foreign particles. And this could be pieces of bacterial cells, viral particles, or viruses, or virions, etc. Right? All kinds of things that the phagocytic cell is digesting, and they're breaking those down into small pieces. Antigens, you can consider a protein an antigen, but most often when we're saying antigen, we're talking about digested pieces, so even pieces of proteins there. Okay, so that's phagocytosis. We will definitely come back to that in just a minute. I'm taking quite a bit of time here.